Hello everyone and welcome to Physics Wallah English YouTube channel. I'm Shalini Somashekar, your botany teacher, and in today's lecture we are going to be covering the entire chapter of microbes and human welfare in one shot under your summary series. As the name of the chapter itself indicates, in this lecture we are going to be talking a lot about microbes in detail. So let's begin today's session by talking about what exactly microbes are. What are microbes? Microbes are also called microorganisms, or in layman terms, people call it germs, right? So what exactly are these microbes? I'm sure you all know what micro means in general English, right? Micro means something that's extremely small or tiny. What could be microorganisms? Microorganisms are those organisms that are super tiny, they're very, very small, too small to be visible to your naked eye. You cannot see them unless you look at them under the microscope. So all of these organisms that cannot be seen with your naked eye and have to be looked at under the microscope are called microorganisms. Now let's talk about its distribution. Where do you think microbes are present? Microbes are present pretty much everywhere on this planet, because of which we say that its distribution is ubiquitous. It means it's found everywhere. You can find it on the land that we walk on, you can find it in the air around us, you can find them on our skin, you can find them within our bodies, you can find them in extremely cold temperatures, in snow, extremely hot temperatures, like in the hot water springs. They are present pretty much everywhere on this planet. So let's take a look at a couple of examples that are given in NCERT. Like I already mentioned, they are present in regions where the temperature can be as high as 100 degrees Celsius. We can't even imagine going close to a place with such a high temperature. I think our bones would probably melt, right? So, but these organisms, microorganisms that have the ability to survive in areas that have such extremely high temperatures are very hardy. They will have their own adaptations to kind of thrive in such extreme conditions. I'm sure you've learnt about thermoacidophiles in biological classification in 11th grade, right? So there you would have learnt about some examples also of bacteria that survive in such high temperatures. Not just high temperatures, there are microbes that are capable of surviving in extremely cold temperatures as well. They can be found even in the deepest layers of snow that is several meters in thickness. Like I already mentioned, you can also find them in the soil, and you can even find them in environments that are highly acidic. So microbes are found everywhere. All right. Now, let's talk about the diversity of microbes. What does diversity mean? What does it mean when we say microbes are diverse? I'm sure you've heard of unity in diversity when we talk about India, right? So we have so many different states and so many different cultures. There is so much variety in India. Despite of it all, we are united. That's what it means. It's very similar when it comes to microbes as well. So microbes are of so many different types. The only thing that unites them all together is that we cannot see them with our naked eye. Unless you put it under the microscope, you will not be able to see them. That is what groups all the microbes together. But in that group, you find so much variety. That is what we mean when we say microbes are diverse. So what are the different kinds of organisms that are included under microbes? I'm sure you've learnt about so many different unicellular organisms in the chapter biological classification in 11th grade, right? There you learn about the 5-kingdom classification, where you have Monera, which includes prokaryotes, then you have Protista with unicellular eukaryotes. So all of these unicellular organisms that are too tiny to be visible under your naked eye, they are grouped under microorganisms. Apart from that, viruses, viroids, prions, all of these are also extremely small. So they also are called microorganisms. So we have bacteria that are unicellular and prokaryotic. Then we have protozoa that are unicellular and eukaryotic animal-like organisms. Then we have fungi, several viruses, viroids, prions. All of these are microorganisms, right? Here is a cute little illustration of microbes having different silly, funny faces. I found it cute, so I put it in here. All right, now let's move on and talk about, how microbes are studied. I already said that microbes are too tiny to be visible to your naked eye, but if you have to study them, you have to grow them in lab, right? You know how there are at least 100 different types of microbes in this room I'm in right now. I'm not able to see them. But if my area of interest is in growing one type of bacterium, one species of bacterium that is probably present on the surface of my skin, I do not want that bacterium or the plate where I'm growing it to be contaminated by other bacteria in this room. So if I have to grow this bacterium, I will have to grow it in an area or in a condition which is very sterile, which is free from contamination of other microbes. So whenever we grow bacteria or fungi or any microorganism for studies in microbiology, what we do is we provide it with food. If they are living in its natural environment, they will know how to get their food. They either synthesize their food or they will have ways to find it from their surroundings. But if I'm growing one microbe of interest, one species that I have to grow, when I'm growing it in the laboratory, I will have to give it food. Because I'm studying those microbes, I will have to give it the perfect conditions to grow so that it will help in my studies, right? I cannot see the microbe, an individual cell, I will not be able to see. But when I provide it with a nutrient medium, what basically happens is we get plates like these that are called Petri plates. So on these Petri plates, you will put the nutrient medium, the nutrients that the microbe requires to grow in a jelly-like substance. You will put it there, and on top of it, you will grow the microorganism of your interest. All right. So all of the nutrient requirements such as nitrogen source, carbon source, all of that you would have provided in this nutrient medium. Excuse me. And you will give it optimum conditions like optimum temperature and everything that is best suited for its growth. Once you provide it with all that and you keep it in sterile conditions in the laboratory, on this plate you will not find any other microbe from that surrounding, and also one bacterium you cannot see. For example, if you're culturing a bacterium, you will not be able to see one bacterium because it's extremely tiny. What happens as the bacteria grows its number of cells will start increasing. One bacterium will start using the nutrient medium, it will grow in size, it will divide and it will become 2, 2 becomes 4, and it will go on dividing. And after a few days or a few hours, you will be able to see dots like these on your nutrient medium. So these are nothing but colonies of bacteria and fungi. Excuse me. So, though microbes cannot be visible to the naked eye, many bacteria and fungi that are grown on nutrient media will form colonies that are very easily visible to your naked eye. And these kind of colonies are used in microbiological studies. All right. So that's one thing that you'll have to remember. Now, I'm sure all of you have studied about a chapter called Human Health and Diseases in 12th grade in your zoology syllabus, right? In that chapter, we talk about so many different microbes that are capable of causing diseases. You've learned about which microbe causes what disease, what are its symptoms, and all of that in detail in that chapter. So basically, in the chapter Human Health and Diseases, you talk about microbes as if they are the bad guys. Generally, common people think that germs are bad, right? Microbes are bad. They're there to cause diseases. They're there to cause infections. But not all microbes are bad. There are some microbes without them, we cannot even imagine our current life. So not all the microbes that exist are villains. There are some microbes that are really, really important for us in our daily lives. We obtain a lot of products from them. So in this chapter, we are going to learn about all the good guys, all the good microbes that we use in our everyday life. And we learn about the exact species of microbes which we use to get certain products. So over the course of this chapter, we will learn about how microbes are important, how they are beneficial, and how they help in human welfare. That's what the chapter is called, right? Microbes in human welfare. So we will learn about microbes and make them the heroes of this chapter as opposed to human health and diseases. All right. So let's begin talking about the importance of microbes in our daily life. Why are you not working? All right. So microbes are used in household products. I'm a South Indian, and in my house, every week, at least twice or thrice, we eat fermented food either as idli or as dosa. And I'm sure in every part of India and most parts of India, dosa and idli are consumed as breakfast or delicacies on a regular basis, right? Also curd, dosa, idli, all of these. To make all of these dishes, microorganisms are required. Let's begin by talking about curd. How do we get curd? I'm sure you know, you may have seen in your households how at the end of the day, whatever milk is remaining, they add a little bit of curd to it, and by next morning, you see that the milk has turned into curd, right? How does this happen? There are certain bacteria in the curd that are capable of converting milk into curd. All right? So, for the formation of curd, not just curd, if curd is formed from curd is where you get your butter and your ghee and all of that, right? So all of these milk products also rely on microorganisms for their formation, right? So there are a group of bacteria that we call lactic acid bacteria. In short, it's abbreviated as LAB. One example for LAB would be lactobacillus. So these bacteria, they commonly grow in milk and they convert milk into curd. What happens during the growth of these lactic acid bacteria is, as the name itself indicates, they are called lactic acid bacteria for a reason. The reason is that they will produce acids, and these acids are able to coagulate. Coagulate basically is curdling. Coagulate and partially digest the proteins that are present in the milk. As a result, milk will get converted into curd. Why is curd important? Curd is very, very important for your gut for two reasons. Number one, it improves the nutrient quality or the nutritional quality by increasing vitamin B12. It is an important vitamin that should be a part of your diet. Secondly, they check the disease-causing microbes. Of course, we know there are microbes that cause diseases. There are microbes that are helpful for us. So what these lactic acid bacteria do, or the bacteria that is there in the curd do, is, in your NCERT it's given that it's present in the stomach, but I'm not sure if it's present in the stomach. I think it's present in your intestine. Okay. So what basically happens is, in the intestine, maybe you could have consumed some food that is contaminated with a disease-causing pathogen. So, as you consume the food, it will go down your alimentary canal and go to the place where these bacteria are residing. These bacteria, the good bacteria, the lactic acid bacteria or the bacteria that's found in your curd, they will prevent those bad bacteria from growing and they will kind of suppress their growth. So, these good bacteria will also help us prevent getting infected from the bad bacteria. So, that is why curd is so important. And also, because of that, your gut health will improve. You may have heard elders in your family say, or you may know it for a fact yourself, how curd is very important, should be a very important part of your diet. That's because it contains a lot of microbes, probiotics, that is very good for the health of your gut. All right. Now, a small amount of curd added to fresh milk as inoculum contains millions of lactic acid bacteria, which at suitable temperatures will multiply. All right. So, in Kannada, we call it heppu. I don't know what it's called in Hindi or in any other language. So, you take a little bit of curd and add it to leftover milk, and that will become curd by the next morning or after a couple of hours. Given the conditions are conducive, the temperature has to be optimum. You may have noticed how during winters, curdling will take a longer time as opposed to summers, because these bacteria grow better when the temperature is a little warmer. All right. So over here what happens is, the little bit of curd that you add to the leftover milk or fresh milk, that is called the inoculum. That will contain lots of lactic acid bacteria, and that will divide and multiply in the milk and cause it to get converted into curd. All right. So that's about curd. Now let's talk about dosa and idli and the microbes that we use in cooking certain dishes. All right. So whenever we have to cook dosa or idli, we make the dough or what we call the batter. After you grind the batter that has rice and some dal and all of that, you grind it and then you let it sit for some time, for a couple of hours. In the next morning or after a few hours, if you look at it, you will see that it has a slightly puffed appearance. The reason why it's puffed is because over here, fermentation will take place. I'm sure you've learned about what fermentation is in your 11th grade in the chapter Respiration in Plants. You learn about anaerobic respiration and fermentation, where whenever fermentation takes place. Okay, so here the batter or dough which we use to make dosa and idli, that also will get fermented by bacteria, and as a result it will produce carbon dioxide gas. So whenever you heat it, that carbon dioxide gas will it will escape, and as it escapes it will leave holes on your dosa. Okay, so I think it's called dosa in some parts of India. In Karnataka we call it dose, and in Kerala I think it's called dosha. So I'm not very sure of its pronunciation. So yeah, so this is dosa and idli for you. If not for microorganisms, we could not have made the delicious dosas and idlis, right? So let's also talk about bread. I think all the Gen Z kids are very, they don't eat dosas and idlis as much as they eat burgers and pizzas and cakes and all of that. So the dough that is used for making bread also it happens by fermentation. And over here, the microbe that is used is a yeast, and that's called Saccharomyces cerevisiae. This is a unicellular fungus. I'm sure you know this, right? So this is a unicellular fungus. It is used in baking industry, because of which it is also called baker's yeast. Now this yeast is available in the form of dried powder like this. You mix it with the dough and you let it sit for some time, and then it'll get fermented, and then when you bake it, when it gets heated, carbon dioxide gets released, which is why our cakes and our bread and all of that will have a spongy and a porous appearance. Okay, so this is baker's yeast for you. Baker's yeast scientific name is Saccharomyces cerevisiae. So far we've only discussed about two microbes names, which is firstly, what was that? Lactic acid bacteria, lactobacillus, and right now Saccharomyces cerevisiae. Over the course of this chapter, you're going to come across a lot of scientific names, all of which you're required to memorize because they are important for your entrance exams like NEET. All right, so that's about baker's yeast. Now let's talk about toddy. Toddy is found in certain parts of South India. I'm from South India myself, so I know. In Kannada, we call this, I think it's called shendi or kallu. I don't know what it's called in other languages. So what they basically do is they will collect the sap from palm trees like this. They will tie pots around palm trees and they will let the sap of these palm trees collect there, which they will allow to ferment, and later you will get a drink that is called toddy. It'll have a small amount of alcoholic content in it. So yeah, that also is obtained from microbes. If not for microbes, this cannot be produced. Now, yeah, microbes are also used in the fermentation of fish, of soybean, and of bamboo shoots in order to make foods. I think this they do in many South Asian countries like Japan. In Japan, whatever fermented soybean you see, I think they call it natto or something. Okay, so it's considered a delicacy in Japan. So for all of these also, we need microbes. If not for microbes, these dishes won't be the same. All right, so thank microbes for existing every single day. Yeah, coming to cheese. Any cheese lovers in the room? Raise your hands. So, I think there are so many different varieties of cheese, right? I don't know much about its varieties, but I know for sure there are a couple of varieties of cheese, and how one type of cheese differs from another type depends on the organism that is used to ferment. Okay, so depending upon that, you get different varieties and different flavors, characteristic flavors in cheese. One type of cheese that I'm very familiar with is this kind of cheese, not because I've eaten it, because I've seen it like 100 million times in Tom and Jerry. You know how Tom always uses this kind of cheese to trap Jerry, right? So this kind of cheese is called Swiss cheese, and the characteristic feature of this Swiss cheese is the presence of these super huge holes within them. Remember what I told you about bread and about dosa? The holes are there because carbon dioxide gets released. For the exact same reason, even in this Swiss cheese, carbon dioxide gets released, which will cause a lot of big holes in the cheese. So this is produced because of large amount of carbon dioxide released, and this kind of cheese is obtained from a bacterium called Propionibacterium shermani. It is obtained from a bacterium called Propionibacterium shermani. Another example that you have in NCERT is Roquefort cheese. Roquefort cheese is mentioned in Peppa Pig multiple times, where it's kind of it has a bluish blackish patches and color on it, and it has a distinct odor associated with it. So this kind of cheese, Roquefort cheese, is ripened by growing a specific type of fungus which gives them a specific particular flavor. All right, so that fungus is I think Penicillium roqueforti. That is the scientific name of this specific fungus that is used to obtain Roquefort cheese. So the name of this fungus is not given in NCERT, so you need not bother. Basically, what makes one type of cheese different from another is the kind of microbe that is used to obtain the cheese. Depending upon that, you get different varieties of cheese with different flavors, different colors, and different taste. All right, so that's all about cheese. Now, let's talk about microbes in industrial products. You know, whenever we make dosa and idli at home, we do it in a small scale. If there's a family of 4 or 5 people, just sufficient for 4 or 5 people, you make that much batter. But there are certain industries that rely a lot on microbes. If not for microbes, those industries cannot even exist. And these are industries that are like $1 million businesses. Can you guess what they are? They are the brewing industry and the production of antibiotics. So alcoholic beverages, and for the production of antibiotics in large scale, if you have to obtain a lot of product, then you have to grow these microbes in an extremely large scale, and you have to grow it in controlled conditions. In order to achieve that, you have huge vessels that are present, which we call fermenters. Within these fermenters, you will grow your desired microbes, and you will have the provision of controlling what temperature you want, what kind of nutrient media you add. If the nutrient medium is used up, how do you remove it? If you need more nutrients, how do you add it? If the product of your interest is produced, what is the way to remove it? You have provision for all of that in these fermenters, and by making use of these fermenters, huge amounts of products can be obtained in a large scale. So microbes are used in industries as well in order to obtain products like beverages and antibiotics. All right, now let's talk about fermented beverages. I told you about Saccharomyces cerevisiae when we spoke about baking industry, right? The same Saccharomyces cerevisiae, the unicellular yeast or fungus, is used in the brewing industry as well, because of which it is also called brewer's yeast. Okay, I'm sure you've learnt about alcoholic fermentation in 11th grade under anaerobic respiration, where pyruvic acid gets converted to ethyl alcohol and carbon dioxide is released, right? So yeast can perform that under anaerobic conditions, and for the same reason they have been used since centuries for the production of alcoholic beverages like wine, beer, whiskey, brandy or rum. So all of these are formed because of the fermentation activity of Saccharomyces cerevisiae or brewer's yeast. Now, what makes them different from each other is the substrate that is used for them to act upon. For example, wine, I think they make use of fruits like grapes, and for, I think, tequila, agave is used, and for vodka, I think potato is used. I'm not really sure about that, but different kinds of substrates are used for these bacteria to act upon, which will make these alcoholic drinks different from each other. All right, so malted cereals and fruit juices, they are used to ferment and produce alcohol. I think barley is also used in the production of beer. Okay, so the substrate will differ, the microbe is the same, the product is different. Okay, so it tastes different depending upon what is the substrate that these microbes are acting upon and fermenting, right? Now, let's talk about wine and beer. I'm sure you've learned in respiration in plants chapter how whenever alcoholic fermentation is happening and yeast is involved in alcoholic fermentation, as the fermentation process progresses, the amount of alcohol in the medium will start increasing. Once the percentage of alcohol in the medium is in between like 12 to 15% or sometimes 10%, the yeast will die. They themselves cannot tolerate that much high amount of alcohol content. So the maximum percentage of alcohol that can exist in a medium is 15%. but you've seen how certain alcoholic drinks have alcoholic percentage over 50%, like 40%. How is that possible? Yeast itself is not involved in producing alcohol of that high percentage. What happens is, after the alcohol is formed, they will put it through fractional distillation. From fractional distillation, you will get beverages that have very high content of alcohol. When you consider wine and beer, these are drinks, alcoholic beverages that have not gone through distillation. So the alcohol content or the percentage of alcohol in such drinks is usually less than 15%. But over here, whiskey, beer, whiskey, brandy and rum, these would have gone through fractional distillation, because of which their percentage of alcohol or the alcohol content will be much higher than 15%. Alright, so these are examples that you'll have to remember, because questions are frequently asked from this in your entrance exams. So this is what a brewery looks like, really cool, like shiny stuff, right? So yeah, that's about brewer's yeast. Now let's talk about antibiotics. I'm sure you've heard of antibiotics. Every time somebody gets a bacterial infection, you go to the doctor, the first thing they do is prescribe antibiotics. Now what are these antibiotics? Let's try to understand it by breaking the word, right? So what are antibiotics? Anti means against, bio means life. Antibiosis or antibiotics means against life. Now what does that mean? Are antibiotics against life means, is it like poison? Of course it's like poison. But is it a poison for us? Definitely not. If it were poison for us, the doctors would not be prescribing them, right? So antibiotics are poison for the bacteria that are causing diseases in us. Alright, so antibiotics kill the bacteria that cause disease in us. Alright, so antibiotics are those substances that are produced by some microbes which have the ability to either kill, they could kill other microbes or retard the growth of other microbes that are known to cause diseases, right? So I'm sure you've heard about certain diseases in the past which took the lives of thousands and thousands of people, such as plague, whooping cough, diphtheria, leprosy, all of these. All of these are bacterial diseases. Because of the discovery of antibiotics, it's become very easy to treat these diseases which were initially considered non-curable. So we must thank microbes for it, right? Now, let's talk about the discovery of penicillin, which is the first antibiotic to have been discovered. It's a very, very interesting story. And this discovery was a chance discovery. It's not like somebody went looking for antibiotics. They did not go looking for whether microbes produce certain substances that we could use in medicine to treat diseases. Nobody went looking for it. People were busy doing something else and they accidentally found that microbes produce certain substances that have medicinal value that can be used in order to treat diseases. How exactly did this happen? This story is beautiful. Look at how it unfolds. So Alexander Fleming, he was working on certain bacteria called Staphylococcus bacteria. He was working on Staphylococcus bacteria in his lab. Like I already mentioned in the beginning of this lecture, microbes are present everywhere. So he was trying to grow Staphylococcus bacteria on petri dishes, okay, for his experiments. For some reason, his petri plate that contained Staphylococcus bacteria, it got contaminated with a fungus. All right. So he observed that a fungus or a mold was growing in one of his petri plates that was not washed. Okay. Okay. So if this is the petri plate, let's say this is the petri plate, he's growing Staphylococcus in it. There is a mold that is contaminating it. Around this mold, this is the bacteria, this is the fungus. Around this fungus, this bacteria would not grow. Around it, it would not grow. So it would grow everywhere else except for the region around it. What he concluded based on his observation is that the mold or the fungus is releasing some chemicals that is retarding the growth of the bacteria. He identified that as penicillin. And the scientific name of the mold that we are talking about here is Penicillium notatum. He observed a mold growing in one of his unwashed culture plates around which Staphylococci could not grow. He found out that it was due to a chemical produced by the mold Penicillium notatum, and after Penicillium notatum, based on its name, he called this chemical penicillin. Here is an illustration, a beautiful image of, you can see how at the center, whatever dot you see here, this dot is of the fungus Penicillium notatum. In the surrounding area, there is a smear of bacteria, Staphylococcus. So this is a bacterium. You can see how there is a zone in the center, right around Penicillium notatum, where the bacterium is not growing. So clearly, this is secreting some chemical that is retarding the growth of the microbes around it. Why would penicillium want to do that? Like I already said multiple times in the same lecture, microbes are present everywhere, and the nutrients that they need are limited. There could be a lot of other microbes growing around penicillium. So, in order to avoid competition or reduce competition for whatever resources, it could be space, it could be food, they will secrete chemicals that will kill microbes, other microbes that are present around it. Because of that, they will get more access to space and to food, right? So that is the reason why they do this. But we make use of that to treat diseases in humans. Not just humans, antibiotics are used to treat diseases in animals as well, right? So though it was Alexander Fleming who first discovered penicillin as an antibiotic, its full potential as an effective antibiotic, was established by two people, Ernst Chain and Howard Florey. After that, it was used to treat American soldiers in World War II. It helped immensely to treat the wounded soldiers during war. It was of high importance to treat many bacterial diseases that had no cure at that time. Because of their contribution, all of these three people, like Alexander Fleming, Ernst Chain and Howard Florey, they obtained the Nobel Prize in the year 1945. Alright, so that's about the discovery story of penicillin. Now let's talk about some chemicals, enzymes and other bioactive molecules that are used, that are obtained with the help of microbes. Microbes are also used for commercial and industrial production of certain chemicals such as organic acids, alcohols and enzymes. There are 4 of them that you'll have to remember. There is a fungus called Aspergillus niger, which is involved in the production of citric acid. Acetobacter aceti, a bacterium, is used in the production of acetic acid. Clostridium butylicum is used in the production of butyric acid. Lactobacillus, which is a bacterium, is used in the production of lactic acid. So over here, for sure you'll have to remember which is the organism that produces what product. You could get questions like which of the following is a fungus that produces an organic acid. So you'll also have to know whether it's a fungus, whether it's a bacterium, what kind of organism it is. Because in entrance exams like NEET, questions can be asked in any way from NCERT. Alright, so it's important for you to memorize all of this. It might seem a little daunting initially considering there are so many names in this chapter, but if you write it all down once or twice and then try to close your eyes and recollect, I think it will become easier. Okay, now let's talk about certain enzymes and bioactive molecules that are produced by making use of microbes. Let's begin by talking about lipases. Every one of you must have watched the Tide ad and the Rin ad and all of that, right? What they do is they basically put something oily on his shirt. They show this oily stain and they put it inside the water and they add whatever soap, powder, detergent. And then what happens? They zoom in and you see the threads in every little, thread of the fabric, and then there are certain molecules that come and they break the oil stain and then they take it out. It's like brand new shirt, right? So what basically they're trying to show there is the lipases. You know what lipases are? Lipases are those enzymes that can break down lipids. So These lipases are obtained from microbes and they are used in detergent formulations. What it basically does is, whenever there are any oily stains on our clothes, the oil can be broken down with the help of lipids and it will clear the stain. So it is used to remove oily stains from laundry. Secondly, we have pectinases and proteases. Both of these are enzymes that are involved in the breakdown of proteins. I'm sure you've noticed the difference between a bottled orange juice and an orange juice that you make at home. If you squeeze an orange and make a juice out of it, look at what it appears like as opposed to a bottled juice of what Maza's mango, no? Any bottled juice for that matter. Compare that with an orange bottled juice. You can see how the bottle juice is much clearer in appearance compared to the orange juice that we squeeze and make at home. So what is causing that difference? Why is it so much more clearer in a bottle juice? Because before putting them inside the bottle, you know for sure that a lot of processes happen in the industries where these bottle juices are getting manufactured, right? So they use these enzymes called proteases and pectinases to break down the proteins that are present in the juice so that it gets a clearer appearance. That's more appealing. It looks more neat, so more people would want to buy it, right? So that's what it's used for. And then we have streptokinase. This is very important. Streptokinase is an enzyme that we obtain from a bacterium called Streptococcus. They take the streptococcus and it's modified by genetic engineering, more about which you learn in the biotechnology unit. This is used as a clot buster. What do we mean when we say clot buster? If there is a clot in your brain and the brain does not get sufficient blood supply, it could result in a stroke. If your heart is not getting sufficient blood, then it could result in a myocardial infarction. So clots could happen anywhere in any part of your body. If whichever part of your body is not receiving blood properly, that part will be affected because blood is what will carry oxygen and nutrients, right? So if at all it is found that some part of your body has a blood clot, in order to break that clot, remove that clot, dissolve that clot, streptokinase is used. So this is basically acting like a clot buster. It removes the clots from the blood vessels of patients who have undergone myocardial infarction leading to heart attack. That is just one case, but even in patients that have stroke, even there they use streptokinase as a clot buster. Now let's talk about cyclosporin A. This is obtained from a fungus called Trichoderma polysporum. This is an immunosuppressive agent. What does this Trichoderma polysporum do? This is an immunosuppressive agent. Where can this be used? Immunosuppressive means it suppresses our immune system. It will kind of calm down the immune system. Why should you calm down the immune system? It's really important. What is the job of the immune system? The immune system kind of has the ability to identify what belongs to your body and what doesn't. The immune system for sure knows what are your cells and what aren't. If there is anything that does not belong to your body, gains entry into your body. It could be something as small as a dust particle, it could be a pollen grain, it could be a bacterium, it could be a virus. Our immune system is super intelligent and it can identify that foreign particle, and what it does immediately, it will start destroying it. But let's say there is a patient in whom both the kidneys have failed. So he has gotten a kidney transplant surgery. He has somebody else's kidney in his body. His immune system will 100% for sure recognize the kidney as somebody else's. What will it do then? If it is somebody else's, the immune system knows one thing to do: to destroy its cells. So at this point, though you know the kidney is not your own, you need the kidney for you to function as an organism. Without the functional kidney, the person will die, right? Kidney is responsible for a lot of functions. It maintains it basically osmoregulates, and it also is a very important essential organ in excretion of nitrogenous waste. So if both of them are not functional and a kidney is transplanted, it is absolutely essential for your body. At that time, what must you do? What can you do? You should ask your immune cells to calm down. When the immune cells calm down, they will not be able to recognize the donor's kidney as a foreign organ and destroy it. Okay, so what happens then is Because you're suppressing the immune system, such people will be more prone to getting other infections. So usually they are advised to stay in isolation, and they are not allowed to visit a lot of people frequently. They are not allowed to get into crowds frequently because they are under immunosuppressive agents. Because their immune system is suppressed, the immune system will not be able to act on other foreign agents as well. So in patients who have had organ transplant surgeries, cyclosporin A is used as an immunosuppressive agent. And also in people who have autoimmune disorders where the immune system kind of can't recognize what is their own cells and what are the foreign cells, it gets confused and it starts killing the body's own cells. In such cases also, immunosuppressive drugs are used. Now let's talk about statins. What are these statins? Statins are produced by a yeast called Monaskis purpureus. In people that have high levels of blood cholesterol, it is used as a blood cholesterol lowering agent. What does it do? It acts by inhibiting the enzymes responsible for synthesis of cholesterol. You know, for the production of anything, for any process to take place, enzymes are involved in our cells. So for cholesterol synthesis also you need an enzyme. What these statins do is they will go and bind with that enzyme and make sure that that enzyme will not synthesize more cholesterol. Okay, so these are some chemical enzymes and bioactive molecules that you will have to know about. Now, let's talk about microbes in sewage treatment. Now, what is sewage? Why should sewage be treated? What is sewage? Sewage is all of that municipal waste that gets generated in tons on a daily basis in cities, right? So what does this municipal waste contain? It contains large amounts of human excreta. There could be vegetable waste, there could be kitchen waste. A lot of these waste is what constitutes the sewage. Sewage is generated even in hospitals where there are patients that are suffering from many diseases. So it can carry a lot of microbes that are capable of causing diseases as well. All of these sewage that's generated on a daily basis in our cities, before they end up in water bodies like rivers, they will have to be treated, because if they are not treated, it will pollute the water body. All right. So in order to kind of reduce the polluting potential of sewage, it has to be treated, made sure that it will not pollute the water body, and then you will have to, let it into water bodies. So whatever treatment happens before you let the sewage into water bodies is what we call sewage treatment. And this sewage treatment happens in two stages. The first stage is called primary treatment. What happens during primary treatment? During primary treatment, there are no microbes involved. Okay? It's during the secondary treatment that microbes are involved. So what basically happens during primary treatment is there is physical removal of a lot of debris by two processes. First is called sequential filtration, and the second is called sedimentation. So what happens here is, you know that sewage will contain a lot of waste material, right? It could contain chocolate wrappers, it could contain plastic waste. So there'll be a lot of floating debris that you find in sewage. Before it is sent for chemical treatment, physically, whatever molecules are floating on the sewage will have to be removed. How do they do that? By a very simple process called sequential filtration. What happens in sequential filtration? You have filters arranged in many rows. The difference between these filters is in their pore size. There are some filters that have a large pore size. There are some filters that have a relatively smaller pore size. So the first filter will have a large pore size. Although probably somebody would have thrown their chappal in sewage. So that is a big substance. It's a big, what do we say, particle. It's a big thing. So it cannot pass through the filter. It will be retained. And then the next filter is slightly smaller. So it goes on, and all of these floating debris will be removed. So it removes all the floating debris. After that, it is passed on to a tank called the primary settling tank. What happens in the primary settling tank is, this is called the primary settling tank. In the primary settling tank, you allow the sewage to stand for some time. You allow it to settle. So if there is any heavy, dense particles like sand, like pebbles, like grit and all of that, it will start settling at the bottom. That way, you can separate and remove out all of these heavier, denser substances that you find in sewage in the primary treatment. Over here, we are not making use of any microbes, they are not getting used up, but we are removing whatever particles we can by physical removal or mechanical removal, we can say. Okay, so now whatever settles at the bottom is called the primary sludge. Whatever remains on top is called the primary effluent. All right. Now, what happens is, so now this primary effluent will get passed on for the secondary treatment. It is in the secondary treatment that microbes are used, and over here we call it biological treatment. You do not add any extra microorganisms over here. Sewage naturally contains a lot of heterotrophic microbes. So those heterotrophic microbes will help in reducing the polluting potential of sewage. So what we do in secondary treatment is you take the effluent from the primary treatment and you pass them into tanks that are called, okay, so this is the primary effluent. This arrow might confuse you. Okay, so you pass this primary effluent into this tank, which is called the aeration tank. What happens here is you're constantly supplying a lot of oxygen and air into the aeration tank, into the sludge, and you will also have some fan-like things that will keep spinning this around so that the air is getting circulated properly. Why is this required? Remember, the sewage will contain a lot of organic matter. Organic matter that is present in excreta, in vegetable waste, all of this will contain a lot of organic matter. Along with that, sewage will also contain heterotrophic microbes such as bacteria and fungi. Now, the greater the amount of organic content in sewage, greater is its polluting potential. So the whole idea of treating this sewage is to make sure that the organic content or the organic matter in sewage is reduced so that its polluting potential is reduced. Now, is there a way we can measure how much polluting potential a water sample has? Yes, there is. We can measure it in terms of what is called BOD. BOD is short for biochemical oxygen demand. Now, what exactly is this biochemical oxygen demand? Let's take okay. So let's take a water sample. Here you have 1 liter of water sample. Within this water sample, you have organic matter. Within this water sample, you also have heterotrophic microorganisms. Now, these heterotrophic microorganisms, for these heterotrophic microorganisms, this organic matter acts as the food. So, these heterotrophic microorganisms will make use of the organic matter as food, break it down aerobically, making use of oxygen, and reduce the content of organic matter in the sewage. How do we measure that? That is in terms of biochemical oxygen demand. Now, in 1 liter of water sample, the amount of oxygen required by microbes to break down the organic matter present in this 1 liter of water sample is what we call biochemical oxygen demand. Oxygen demand obviously is talking about oxygen, right? How much oxygen is required by the microbes that are present in this sample to break down the organic matter present in this sample. Obviously, if the organic matter is more, if its quantity is high, you will need more oxygen. So, greater the amount of organic matter, greater is its polluting potential, greater would be its BOD. As these microbes that are present in the sewage will start making use of oxygen and breaking the organic, using the organic matter, the organic content here will come down. The number of bacteria and fungi will start increasing. As the organic content will come down, its BOD will reduce, its polluting potential will reduce. So what basically happens during secondary treatment is the organic matter content in sewage will decrease. Making use of this organic matter as food, the fungi and bacteria that is there in sewage, they will start growing and increasing in number. Eventually, BOD will reduce. As the organic content reduces, obviously BOD will reduce. As BOD reduces, polluting potential is reduced. So ultimately, once the BOD has significantly reduced, the effluent will be safe to be let into water bodies. But before that, what happens is, over here, the bacteria and fungi would be just hogging on all of this organic matter and growing like in crazy amounts. So it will start forming filaments like this. The fungi, you know, hyphae will be there, right? Fungi body is made up of thread-like structures, filament-like structures that are called hyphae. So they will start increasing in number, and also a lot of bacteria will multiply, and it will form structures that are called flocks. As the flocks are getting formed, organic matter in it will reduce. Eventually, you will allow these flocks to settle down. Once these flocks settle down, okay, so once these flocks settle down here, we call this settled flocks as activated sludge. What we do with this activated sludge is you will remove it. A portion of it you will add as inoculum to the primary settling tank. Remaining activated sludge is passed on to tanks that are called anaerobic digesters. In these anaerobic digesters, there are certain microbes that will, without making use of oxygen, break it down and produce biogas that can be used as a fuel. Okay, more about it we'll learn going forward. For now, just remember, at this point, the flocks will form by making use of the organic content in the sewage, and you will allow it to settle down once the BOD has significantly reduced, and then you will remove it out. Whatever remains, the effluent, that will be safe to be let into water bodies because it will not have a very high polluting potential. So that is about secondary treatment. In some sewage treatment plants, you will also have tertiary treatment where you treat this effluent with chlorine and UV rays, but that's not given in your syllabus. So don't worry about it. Just talk about primary treatment where the sequential filtration, sedimentation, secondary treatment where heterotrophic microbes that are naturally occurring in sewage will act on the organic matter and reduce its content, bringing BOD down and also bringing its polluting potential down. All right. Now, let's talk about some initiatives that the government of India has taken to save our rivers, right? So, the Ministry of Environment and Forests have initiated certain plans like Ganga Action Plan and Yamuna Action Plan to save these rivers from getting polluted. So what basically is the agenda of these plans is they plan to construct a lot of sewage treatment plants to make sure that the sewage that is let into water bodies is treated before it goes into the water bodies. That way you can reduce the pollution that we see in rivers, major rivers of India like Ganga and Yamuna. All right. Now let's talk about production of biogas. I already said that a large portion of activated sludge that we get in the secondary treatment is pumped into anaerobic digesters where certain microbes will act upon it and produce biogas which can be used as a fuel. So what is this biogas? It is a mixture of certain gases such as methane, carbon dioxide and hydrogen. So it contains methane predominantly. It is inflammable, so it can be used as fuel. So how is this biogas produced? Why do we call it biogas? Because some living organisms are involved in producing it, and it's usually produced by certain microbes. So that, like I already mentioned, can be used as fuel. So the type of gas that is produced will depend on the microbes that are involved and what is the organic substrate that they utilize. You've learnt about methanogens under archaebacteria in biological classification in 11th grade, right? So what are these methanogens? Methanogens are those bacteria that anaerobically grow and they generate methane. That's why they're called methanogens, right? So these bacteria will grow anaerobically on cellulosic material. Cellulose is present where? It is present in the cell walls of plant cells, right? So it will grow on cellulosic material and it will produce a large amount of methane along with carbon dioxide and water. One example for a methanogen is methanobacterium. So where do you find this? You can commonly find this in anaerobic sludge. Okay. So apart from that, you can also find it in the dung of cattle, in cow dung, in cattle dung, you can find it because cattle, they feed on plants. So plant cells have cellulose. Since they themselves cannot digest cellulose, in the alimentary canal of these cattle, there is a organ that we call rumen. Within the rumen of these organisms, methanogenic bacteria will be present. They will act on the cellulosic material and will produce methane and carbon dioxide and water. All right. So a lot of cellulosic material present in the food of cattle is also present in the rumen. These bacteria help in breakdown of cellulose and play an important role in the nutrition of cattle. So this excreta or dung of cattle, which we call gobar in Hindi, that contains a lot of these methanogenic bacteria. So the cow dung can be used to generate biogas, but because it has a high amount of methanogenic bacteria. So what does this biogas plant look like? It has a concrete tank. So this is the concrete tank right here, which we call the digester. So this concrete tank is about 10 to 15 feet deep. All the bio wastes are collected and fed into this digester in the form of a liquid that we call slurry. So there is an inlet to feed this slurry. On top of this digester, there is a floating cover that we place here. As you can see here, there is a cover that floats on top of the digester. This will keep rising as the gas is produced within the digester. We have two outlets. One outlet is connected to a pipe. This will supply the biogas to households that are present in nearby the digester. There is another outlet over here through which whatever slurry is used up can be removed, and that can be used as manure in the fields. Usually cow dung is present in available in large amounts in rural areas. So that is where you mostly see the biogas plants. You don't see them in cities, right? So what can this be used for? It can be used for cooking and lighting in nearby households. All right. Now let's talk about, which are the industries that are involved, which are the institutes that are involved, organizations involved for the development of biogas production in India. One is Indian Agricultural Research Institute, which is called IARI. Another one is Khadi and Village Industries Commission, which is called KVIC. This you have to remember. Now let's talk about microbes as biocontrol agents. What is biocontrol first? Biocontrol refers to the use of biological methods for controlling plant diseases and pests. Usually what we do, we spray pesticides and insecticides to get rid of insect pests in crop fields, because if they cause infection in plants, that will reduce the yield, right? So they may be, these chemical insecticides, weedicides and pesticides, they may be helpful in getting rid of the weeds. But there are some demerits associated with them. That is, they are toxic and extremely harmful to human beings and other animals. Secondly, they pollute our environment. They pollute the soil. It will leach into the soil and it will affect the quality of the soil, and also the groundwater quality will be affected. It will pollute the environment. So because of this, there are other ways we can come up with, where we make use of living organisms, some other living organisms, in order to get rid of pests and insects that cause a drop in the yield. All right. So what this basically relies on is natural predation instead of artificial chemicals. What an organic farmer believes is that biodiversity furthers health. See, these chemical insecticides and weedicides that they spray in fields, along with spoiling or getting rid of pests that cause infections and infestations, they might also get rid of useful animals and insects. You know how several insects can be pollinators, right? So certain times the chemical insecticides and pesticides that are sprayed can get rid of the useful ones as well. So that is a demerit. So what organic farmers do is they rely completely on biocontrol and they do not spray pesticides, insecticides and all of that. So if you have to eradicate creatures that are described as pests, it is possible, it is undesirable, it is not good to do that, because let's say this is a plant, this is a pest, that's a very silly diagram of a pest, and this pest will cause this crop yield loss, and there is another organism that depends on this pest. If you eradicate this, this could also go extinct because this depends on this. Either this could feed on this, or there could be some symbiotic association, or this could be a parasite on this. This basically requires this to function. If you get rid of this, even this can get lost. Okay, so that is a risk. So insects that depend on them as food or host would not be able to survive. As we start using biocontrol agents, our dependency on chemical pesticides that are very toxic will start getting reduced. So what farmers basically need to be aware of in order to use biocontrol agents is the life cycle. Life cycle of the insects that are acting like pests in their field. So if there is a pest that is acting as, if there is an insect that is acting as a pest in your field, if you know about its life cycle, if you know about what are its natural predators, then it's easier for you to manage your field without using any chemical pesticides, right? So, yeah. Here are some examples. Though this chapter talks about microbes, here you have examples of certain insects also that act as biocontrol agents. Ladybird beetle, I'm sure everybody knows this. It's considered like a lucky symbol where you have a red colored body with black color spots on top of it. That is the ladybird beetle. It's used to get rid of aphids. Dragonflies are used to get rid of mosquitoes. There is a bacterium called Bacillus thuringiensis, which is called Bt. This is used to get rid of butterfly caterpillars. You know how these butterfly caterpillars, they will feed on the leaves and then they form a cocoon. So the crop loss can happen because of butterfly caterpillars. If you want to get rid of them, you can use this bacterium called Bacillus thuringiensis. These bacteria are available in the form of dried spores in sachets. So you mix them with water and spray them on the vulnerable plants, and whenever the caterpillars or the larvae feed on it, they will die. Okay, so they've made use of Bacillus thuringiensis in genetic engineering also in order to produce plants that are naturally resistant to certain insect pests. One example for that is Bt cotton. More about how exactly it's produced, what are the genes involved, all of that you learn in your biotechnology chapter. Okay, so what are some other species? Trichoderma is a fungal species. They are free-living fungi that are commonly found around the roots of plants. So this also is known to control several pathogens that cause infections in plants. Baculoviruses. Who thought viruses could be useful? Baculoviruses are a class of viruses that are used as biocontrol agents. So usually these viruses attack insects and other arthropods. So if there is an insect pest that is a cause of worry in your field, you can make use of these viruses. Usually it is the genus nucleopolyhedrovirus that is used in biocontrol. So like viruses are extremely species specific. What viruses cause infection in plants? They do not cause infection in animals. So if there is any virus that specifically targets insects, even if you consume it, it will not be a problem, right? So yeah, there is no negative impacts on plants, mammals, birds, or even on non-target insects. Now let's talk about microbes as biofertilizers. Similar to how pesticides and weedicides are chemicals that are used in crop fields, we make use of fertilizers as well. What are fertilizers? Fertilizers are chemicals that you add to the soil. It could be in the form of urea, potassium, nitrogen, all of these fertilizers you get. So these again are chemicals and they could be harmful for our health, they could be harmful for the soil, they could be harmful they could pollute the groundwater, so they are bad also. So how do you avoid that? By making use of biofertilizers. Biofertilizers are living organisms that have the ability to enrich the fertility of the soil, or basically the nutrient quality of the soil. The main sources of these biofertilizers are bacteria, fungi, and cyanobacteria. There are some examples. Let's take a look. Bacteria. You have rhizobium. I'm sure you've heard about rhizobium. Rhizobium is a rod-shaped bacterium that you find in the soil. They have symbiotic associations. They develop symbiotic associations with the roots of leguminous plants like peas and beans. And over there, they help in nitrogen fixation. In return, they will get food from the plant. Okay. So azospirillum and azotobacter, these are also nitrogen fixing bacteria, but these are free living. They are not symbiotically associated with any plant. They are found free living in the soil and they help in nitrogen fixation. Talking about cyanobacteria, cyanobacteria are photosynthetic bacteria. Some examples are Anabaena, Nostoc, Oscillatoria. They are involved in nitrogen fixation as well, and many a times they are used as fertilizers, biofertilizers in paddy fields. Coming to fungi, fungi are known to form symbiotic association with the roots of higher plants like gymnosperms and angiosperms, where they form mycorrhizal associations. Because of the mycorrhizal associations, the roots of the plants will have the ability to absorb water and minerals better from the soil, specifically phosphorus. They will help in enhanced absorption of phosphorus from the soil and pass it on to the plant. One example for that is the genus Glomus. The fungal genus Glomus is known to form mycorrhizal associations. So, apart from allowing the plant to absorb water and minerals better, they show several other benefits such as resistance to root-borne pathogens. If there are any pathogens that gain entry into the plant via the root, they will resist those pathogens as well. They will have a better tolerance to salinity and drought and overall increase in the development of plant. Okay, so that's about biofertilizers. I hope this is clear. So with this, we have come to the end of today's session. You saw how microbes can be such saviors and heroes in so many cases. So the next time you hear about microbes or germs, don't think they're all the bad guys. So in this chapter, you have a lot of things to memorize. I know it can seem a little overwhelming, but trust me, if you practice it like a couple of times and try to recollect it and revise it frequently, it won't be a big task for you. I hope this session was helpful. So with this, I'm going to sign off from today's session. Until I see you next, don't forget to keep revising. Stay healthy, stay happy, and keep smiling.